Great. Next up, we have Justine Hansen from McGill University, who will talk about spatial nulls.
Cool. So the clicker doesn't really work. I should use the mouse, maybe? Let's see my mouse. Okay. We'll see. Okay, so yeah, I'll be talking about spatial autocorrelation. Let's just start with what that is. Maybe you guys are already using this in your work, but in case you aren't, I'm hoping that like no matter how much you know, you will learn something. So let's just start with what is spatial autocorrelation. There's this like first law of geography that says that everything is related to everything else, but near things are more related than distant things. And this applies to not just geography, but also neuroscience, not just neuroscience, but also like life in general, honestly. So it's kind of a good rule to go by. And in the brain, how you might visualize this is that brain regions that are close together tend to be more similar than brain regions that are far apart. You don't have to take my word for it. You can look at this in lots of different data sources. So for example, here's a similarity matrix of how similar brain regions are in terms of the genes they express. And that's going to decrease with distance. So regions that are far apart are less similar in terms of the genes they express. But not just gene expression, all sorts of different features. This is something we see over and over again. We kind of have to take into account this fact that we see in the brain because it is spatially embedded, it exists in space. Regions that are far apart tend to be less similar than regions that are close together. Okay, but why do you need to care about this? I'm going to try to come at this from a few angles, but one reason, spatially naive models, so statistical models that don't take into account spatial autocorrelation, they tend to assume that brain regions are independent and identically distributed or exchangeable. And so the idea here is that if you have two observations, in our case it's a brain, so two observations would be two brain regions, we need to be able to swap these and assume that we haven't like disrupted any relationships. And if you don't care about distance, then maybe you can do that. But in the case of the brain, there is a relationship between those two brain regions. In fact, there are many relationships, but one of the ones that we're going to focus on here is just distance. And so if you swap two brain regions that are close together versus two brain regions that are far apart, that's a very different swap that's happening, right? So we want to be able to take that into account. Here you can see that the probability of having a statistically significant correlation between two brain maps is going to, if you use a spatially naive model, you're going to have real statistically significant, I mean, when I say real, I just mean like very, very small p-values. Whereas if you take a null model that accounts for the spatial autocorrelation, you're going to control for that, or maybe not control, but you're going to have a null model that accounts for the fact that your brain maps are spatially autocorrelated, and so your p-values are a bit more. Let me see if I know how to use the laser. Which button is that? Here, these guys. These are the spatial autocorrelation-preserving null models. The p-values are not going to, or the probability of having statistically significant p-value is not shooting up as much. You can see it's a bit better conserved. Whereas this guy, this is spatially naive. The more autocorrelated your brain map is, you're going to get this really, really small p-value, even though that could just be because of spatial autocorrelation. Third way I'm going to try to motivate this, here are two brain maps that are completely random. This is simulated data. By the way, I didn't do this. This is coming from Markello and Misick 2021. And then we're going to tune the autocorrelation of these brain maps ever so slightly. We're just going to keep tuning that autocorrelation until their correlation, the correlation between these two brain maps is massive, negative 0.9. So if you saw a correlation negative 0.9 in your data, you might be like, that is awesome, I'm going to publish a really awesome paper about this great correlation. When in fact the underlying data is actually completely random, their correlation, their true correlation is zero, it's just their spatial autocorrelation is like jacked way up. So for example, let's say that one of the brain maps was like degree, how many connections do you have? The other brain map is metabolism, how much are you, let's say, processing O2? Obviously that's not what these are, but just as an example, you might be wondering, are brain regions that have a lot of connections, are they also the brain regions that metabolize a lot? Are they more energy expensive? And you're going to want to understand if that correlation or that relationship survives this property of spatial autocorrelation. Okay, so I'm going to go into the theory a little bit about spatial autocorrelation just so that we understand what it is, how do we measure it, and then from there build an intuition of how we also control for this or build a null model that preserves spatial autocorrelation. Here's a brain map. The question here is, do spatially proximal regions tend to have similar measurements? So even the nature of that question, you can tell that like the value, we're going to end up with a value here. We're going to end up with some number that tells me how much is that the case? How much is it the case that spatially proximal regions tend to have similar measurements? So let's say we have some xi up there. We want to know if some other brain region, do they have similar measurements or not, and does that depend on distance? Okay, so what we're going to do is we're going to loop through every pair of brain regions. So for every xi and every xj, we're going to say, how much does this guy deviate from the mean? How much does this measurement deviate from the mean? And how much does this guy's measurement deviate from the mean? And then we're going to multiply those two together. So how much does the measurement in xi deviate? How much does the measurement in xj deviate? Take the product. Okay, so if we take the product, that means that if these two are both deviating a lot in the same direction, they're both way above the mean or way below the mean, take the product, that's going to be positive, right? So you can already get a sense that this question, the way that I've asked it, do spatially proximal regions tend to have similar measurements? If it's yes, then we're going to have a big value here, right? This is going to be positive, that's going to jack up my sum. And if they're very different from each other, this is going to be negative, that's going to make my value smaller. Now, very importantly, this is spatial autocorrelation. So correlation, this is pretty much what a correlation is. You take the product of your measurements compared to the mean. Auto, because it's the same, instead of x and y, it's xi and xj, so it's the correlation of you with yourself. And spatial is like the key word here. We have a weight of distance. So in this case, we want the two regions that are close together, we care more. So we want that value to be bigger, so that's going to be something like inverse distance. And which distance matrix, or how you make your distance matrix, there's a lot of different ways you can do this. But let's say like inverse Euclidean distance, something like that. Then we're going to normalize this by the total variance in the brain. So this is also the same as the correlation for, guys, don't take pictures yet, there's more of the equation. Yeah. And we're going to normalize this value so that it ends up between -1 and 1. This normalization is just the number of regions divided by all the distances in that weight matrix. Yeah, now you can take pictures. And what's nice about this is because it's normalized between -1 and 1, it's very interpretable the way a Pearson's correlation is. So this value, which I'm calling I, is called Moran's I. It's named after this guy, Patrick Alfred Pierce Moran. He's down there in the corner. And like I said, it is... Oh, sorry. How do we go back? Yeah, so let's just walk through this. If we have these two brain regions... Is there any way I can do this with a mouse? Not really, okay. If we have this brain region and this brain region, they're very close together and their values are very similar. So this is going to say, like, when I compute this part of the sum, this is going to be yes, regions that are close together, they're very similar. But if you take these two brain regions, where xj is now kind of orange looking, their values are a bit more different. So this product is going to be either negative or relatively small. But the thing is, we don't actually care because they're so far apart, so they're not going to be weighted very much. So I don't care so much that these two regions are very different from one another. But then these two brain regions, they're very close together and very, very different. So that's going to give me a large negative value here. This weight value here is going to be pretty big. And so that's going to make my I come down quite a bit, right? And you can sort of develop an intuition for the fact that Moran's I, if you have an I of 1, this is going to be perfectly spatially autocorrelated, so regions that are close together are very much going to be similar to each other. So your value is going to depend a lot on the values of your neighbor, whereas I of -1 would be that your value is completely opposite of that of your neighbor. Think like a chessboard, like that checker pattern. That is the spatial autocorrelation of -1. And autocorrelation of 0 is going to be that it's pretty random, so your value does not really depend at all on your neighbor's values. Yes, this is a global measurement, so you get one Moran's I for the entire brain map. And it's very similar to a Pearson's correlation in definition. So in a Pearson's correlation, if you remember, instead of taking your value at region I and region j, it would be for two different observations, right? So you'd have like x minus mean of x and y minus mean of y. But autocorrelation, so it's the same guy. It's very interpretable, and it is Australian, because Patrick Alfred Pierce Moran, such a great name, is an Australian. Okay, oops, wrong button. So, second way, how do we calculate spatial autocorrelation? That was a way to give you one global measurement for the entire brain map. Now I'm going to give you a slightly different question, a different value. This is now answering the question, how do measurements vary as a function of distance? So this is no longer going to be one value for the entire brain map. This is going to be a graph. It's going to be a function that changes depending on how far apart the measurements are. So let's say we have some distance, h. Let's say it's this guy. The question here is, how much do my measurements vary when my distance is h? So I'm going to look at, maybe my xi is over here and my xj is over here, but then if I keep h the same, I'm going to take a look at all those regions with distance h around this xi, right? but then we're going to do this for all the xi. So I'm going to call this some variation as a function of h, the distance, the measurement variance when samples are a distance h apart. I'm going to loop through all those xi's. And I'm going to say, what is my measurement at my xi distance h away? So in this case, xj over here, multiplied by, clicker is not working, give me a second. There, multiplied, or sorry, take the difference of what my measurement is at xi. So pretty much, what is my measurement h away? What is my measurement where I am? What's the difference? How much am I deviating from where I'm standing? Take the square of that because we don't care about sign, and then take an average. And this is going to tell you how much do my measurements vary at this function h. And so if you start with a really short distance, so h is really small, your variation is, if in a spatially autocorrelated system, your variation is going to be quite small. So you're going to see that if you're with a small h, all these measurements are quite similar to one another. As we make h bigger and bigger, your variation is probably going to get bigger. And at some point, your variation is going to kind of plateau. Because at some point, it doesn't matter how far away you are, you do not have any impact on me anymore. You know what I mean? So I'm going to be, like, my measurement is going to be impacted by those that are close to me, a little bit impacted by those that are a little bit further away, and then at a certain point, it doesn't make any difference. And this is called a variogram or semi-variogram. It comes from this guy, Georges Moran. He's French. There he is, being very busy. Okay, so that was two ways of measuring spatial autocorrelation. Sorry. Two ways of measuring spatial autocorrelation. First way, Moran's I, that's a global measurement. Second way, this variogram, that's a function of distance. Now, how do you account for this spatial autocorrelation in your statistical analyses? We're going to want to make a null model. There are different ways of doing that. I'm going to go through all of these, and then let me just first, I don't really have to tell you this because the previous speakers have already told you, but like the whole point of making a null model is that you have some sort of real relationship, your empirical relationship, the thing that you're interested in. It doesn't have to be a correlation, it's just correlation is the easiest thing to talk about, so that's my example here. But you have some real correlation, you're going to want to compare, as other previous speakers have said, how surprising is that compared to a null? And so we're going to make a bunch of bogus maps. These are going to preserve the spatial autocorrelation of my original map. You're going to calculate a bunch of correlations with those maps. Not working again. There we go. You're going to have a null distribution of your null correlations. You're going to have your empirical correlation, and you can see whether or not you beat the null. In this case, you did not. P equals 0.06. Sucks to suck. Anyways. So that's why you're going to want to be able to develop this null. And so what I'm going to talk about, go back. No, it does not work. What I'm going to talk about here is how to create these null brain maps. Okay, so the first one I'm going to talk about is the spatial permutation model, or the spin test. So maybe you guys already know, but it's totally fine if you don't. The idea, the just kind of like big, broad, big picture concept here is that we have a brain map. We're going to project it to the sphere, which I know that's already a little bit like, I'm just going to, just trust me that we can do this, We're going to spin that sphere around and then plop the way that the data has been spun back onto the cortical surface. So I'm going to wave my hands a little bit. You have a brain map, project a sphere, you rotate, put it back down on the brain. And so your regions have been moved around a little bit, right? But your neighbors are all the same. So the spatial structure of this data is totally the same. It's just that instead of maybe being from front to back, now this brain region is at the side or something like that. And then you do this a whole bunch of times. You don't just want one null, you want like 1,000 or 10,000 nulls so you can create this null distribution. So I'm going to walk through these steps and give you a little bit of like history of what's going on here. So this method, the spin test, actually, I don't think it was called the spin test in the original paper, but at any rate, this method, it was the first time it was formally like introduced as a paper is in this Alexander Block paper 2018. It's not that long ago. But the first time that it actually showed up, or like the first place that these ideas started to show up is in this OHBM abstract. So this was never actually published, but there's an OHBM abstract from 1996, which is like nearly 30 years ago, by Marty Sereno, where he starts to talk about or have this idea of being able to project the cortical surface to, in this case, it's an ellipsoid. And so that this entire method, the spin test, it totally depends on being able to do the spherical projection. And there was no way you could do a spherical projection before people started having the idea of projecting it to an ellipsoid. And then a couple of years later, Bruce Fischl came in and published. By the way, Marty is a co-author on this paper as well. This is a very famous paper. You've probably cited it in your work. But here, they scrap the idea of the ellipsoid and make it a sphere, which is way better, because everybody much prefers spheres than ellipsoids. Ellipsoids are just kind of a pain to work with. And so because of this, we can do our spherical projection. And then in 2013, Alexander Bloch, same guy who put out the kind of formal description of this method, 2013, he was just doing his own analysis, and then at some point in the analysis wanted to have some fake versions of his modules that he created. And the way that he does it, he calls them pseudo modules. The way that he does this is actually to do this rotation. He projects it to the sphere, rotates it, but at this point doesn't call it like a spatial null or anything. He just says, oh well, that makes sense. That's a way to do something, so I'll just do that. This is like not important. You don't have to know this. So I'm just giving you the history, the background of where this method came from. 2015, a couple years later, somebody else also used this method, but a bit more clearly, at any rate, it's a bit more, it's a whole figure in the paper about taking the spherical projection of the surface and then changing the origin point. So here they start to talk about this rotation method. It's coming up more and more. And then in 2016, there's also this paper here. They don't have a figure, so I can't really show you exactly what it looks like, but they say very, very clearly, we rotated each hemisphere of the original parcellation a random amount around each x, y, and z axis of the spherical expansion of the 32K epicellar surface. So they took the brain, projected it, it's on epicellar, projected it to the sphere, and rotated that. And that's how they built their null. Okay, so now at this point of the talk, I'm going to zoom in on this idea of how in the world we do the rotation. So I have talked about the spherical projection and kind of given you the history of that. I'm now going to talk about how is it that we do that rotation. I'm going to be totally honest with you. I can't click next. I personally think this is very cool and interesting. However, it will have zero impact on you or your life or your research. So if you need a time to tune out, this is the time to do it. Okay, let's just remember how polar coordinates works, right? So if you have a sphere, you can pick any point on that sphere with two angles. You just need one angle that goes around the equator, that we're going to call phi, and one angle that goes from north to south, that we're going to call that theta. Okay, so let's say I have a sphere here. I want to pick this point over here. I need to know where I am around the equator. I need to know where I am north to south. You guys with me? Nods. Nice. Okay. Phi is going to go between negative pi and pi. It goes all the way around the circle, so that's the equator. Theta, it doesn't have to go all the way around. Again, we only need to go north and south. So it's going to go pi over 2 to negative pi over 2. And this sampling space of these two angles to pick a random point on the sphere is going to look like a rectangle. So we have phi going all around the equator, negative pi to pi, that has a length 2 pi, and theta going from pi over 2 to negative pi over 2, that has a length pi. So it's a rectangle, right? And if you can imagine, when you take a sphere and rotate it, another way of thinking of that is just saying that you've picked a new origin point. Right? And so what I'm going to do is I want to pick a random point in the sphere. That's going to be my new origin. That's how I do my rotation. So you can think all you need to do to pick a random point on the sphere to do this rotation is to pick a random theta and pick a random phi. And then your job is done. And that's actually what they did originally in that 2018 paper where eventually was nodding because anyway, he knows exactly all about this. That's what they did in the original paper. They picked a random theta, they picked a random phi, and they said, this is, that's my new origin point, that's my random rotation, and I'm done, I don't need to worry about anything. However, if you imagine we have a few points close to the equator, okay? So close to the equator means that theta, the thing that goes from north to south, that's going to be close to 0, right? Close to the equator. So you can imagine these points over here are going to correspond to some points close to theta equals 0. It's going to look something like, it's totally killing the timing of my jokes, not that I have any jokes, but anyways, okay. So we have some points on the sphere here, close to theta equals 0. That's going to be sampling space around here. Okay, so let's imagine something like this. We're going to have points. The only reason I'm not going past phi 0 is because I'm pretending that these points are on the outer edge, on the inner edge, but it doesn't really matter. But in any case, we have some points that we've selected on the sphere. They're random. We have randomly sampled. Now, I just want you to watch this right here. What happens when I decide to move my points from the equator to the pole, okay, to the North Pole. the sampling space of my rectangle has not changed at all. This is uniform sampling still, right? So I have the same number of points close to theta equals 0 as I do close to theta equals pi over 2. That's the North Pole, which is good for the rectangle. That means I'm uniformly sampling in the rectangle. But on the sphere, what this means is that my points are now in a way smaller area. Okay, so before I had these points spread out along theta equals 0. Now all my points have been condensed into this pole, and the reason it's doing this is that uniform sampling on a sphere, uniform sampling on a rectangle. Here we go. Uniform sampling on a rectangle does not equal uniform sampling on a sphere because the surface area matters. So just because you're picking a random theta and a random phi does not mean that you're going to randomly rotate a brain map or randomly rotate a sphere, because you're not going to be randomly picking a point on a sphere, because you're going to have way more points in the poles than you are going to have around the equator. Okay? Now, this is like very awesome and cool, but really does not matter to you, but still I just wanted to tell you because I thought it was cool. Here's an awesome animation that Jiangxi, another PhD student in the lab, made. Just gives you a sense. Here, uniform sampling in the rectangle, so you are same frequency of points no matter where you are in this rectangle, but on the sphere, it is not uniform at all. In the poles, it is much more condensed. You're going to have way more rotations in the pole than you do in the equator. Now, by the way, so the kind of story of this, well, first let me just tell you that you don't have to worry about any of this because it has already been accounted for in the code. The way that you manage this is instead of picking two random points, you do something that's called a QR decomposition. This is a linear algebra method that takes your matrix and decomposes it into two other matrices, one of which is a rotation matrix that does ensure that there's uniform sampling on the sphere. I'm not going to go into the math. Honestly, I don't know the math, but you're just going to have to trust me on that. In the original 2018 paper, Alexander and Bloch present this spherical spin test thing, right? They projected the sphere, they rotate the thing, pop it back down on the brain. At this point, they actually did not deal with the whole uniform sampling issue. It was just a few months later that this other paper, here, Lefebvre, 2018, they do essentially the spin test, and they say in their text, by the way, like our method is a bit better than the Alexander-Bloch method because we do manage this uniform sampling issue. And then Alexander-Bloch, like right after that, said, that's true, good point, we did not realize that was the case, so we're going to update our code, so now our code takes into account this random sampling of the sphere, so it includes the QR decomposition. Thank you very much. And I don't know, this is just like a cool example of how, when you do science, you don't have to be perfect at the same time because someone else is going to tell you how to make it better, and then you go back and make it better, and then everybody succeeds. So now you don't have to worry about it because it's already implemented everywhere that you do the spin test. I just realized I have not mentioned, but here at the bottom, throughout this talk, I'm going to have the actual function that can do this for you, as well as alternative or original codes. For example, this is a Python version of the MATLAB version down here. Okay, so that was the spin test. We have our brain map. We know how to project it to the sphere. Thank you, Marty Soren in 1996. We now know how to spin it, thanks to random sampling on the sphere and that whole thing. And then now we want to pop it back down on the cortex, and to do that we have a few choices, because there's a lot of different ways that you can put data on a sphere back down onto data on a cortex. Just imagine, like, that's a brain region here. What does that correspond to? Like, how am I supposed to decide which parcel in the brain to put it back onto? Now, whether, the different versions, there are many different versions of doing this, and there's no right one. It depends a little bit on your situation. And the reason there's so many different versions is that this original 2018 paper described the spin test for vertex-level data. But then a lot of people wanted to apply this to parcel-level data. And then so, like, 4 different people kind of independently came up with unique ways of doing that, and so now we have a bunch of different ways of doing this, which is not a bad thing at all. So here, I'll just go through this and show you kind of all the different versions. So depending on what you want to do, there are different ways of popping that data back down on the cortex, and the ways in which they are different are, for example, you have some of these versions here that may allow duplicates. So you can imagine when you project the sphere and then rotate, let's say that you want to put your spherical parcel back down on the cortical parcel, and you just want to minimize the distance. So if you do that, you might have 2 rotated parcels that are equally distant, so you're going to have, let's say, 2 rotated, that parcel is going to show up twice in your rotated brain map. And maybe that's fine. If you don't care about the distribution of your values, or you don't mind that every single parcel shows up exactly once in your rotated version, then that is totally okay. You can do these these versions. However, if you do want that every value only shows up once, which, so for example, if you're doing network-y things where you care about the degree distribution, or you just want that every value shows up exactly once, then you might want to do these two versions down here. They take a little bit more time, but in my experience, not too much time. Like I've used both of these and it has been fine. Also, you will have some of these that will allow the medial wall to come into the brain. So I haven't mentioned, but the spin test, it does this for one hemisphere. It doesn't do this for both hemispheres. So you do it for each hemisphere separately, and you can imagine the one hemisphere, there's a bunch of that hemisphere at the medial wall that you don't actually care about. So when you project this to the sphere, you have this whole like gray patch, and then when you spin that, you have a whole gray patch that ends up back in your brain, not unlike these gray patches. And in some cases you don't care, those are just nands, and if you're doing a correlation, you just skip those brain regions, and that's fine. But maybe you do care and you don't want to have the medial wall end up in your cortex, and you just avoid these. Now another thing is this works for vertex level data, and the rest are parcellated data only, so that also depends a little bit on your situation. All of this, by the way, is in this paper here, Marcello and Missick 2021. And then big caveat, all of this, the spin test in general, remember the spin test depends on the spherical projection, and that spherical projection only works for the cortex so far, right now, the cortical surface. And so that means that this entire process of spinning your data around only works for cortical surfaces. Okay, so what do you do if you can't rotate your brain map? We're going to generate a new map. And people have already talked about this now, so you guys are kind of pros, but you know that you can either take your empirical data and shuffle it around some way, so that's the spin test, right? You're shuffling it in this very specific way, in other words, a rotation. Or you can generate new data, that's the generative method. So, wrong button, does not work. Okay, so in this case, we have some brain map, and it has variogram. Remember the variogram? So that's the function that depends on distance, and it tells me about my spatial relationship in my data, this function, distance. And so your brain map is going to have some variogram, and then what we're going to do is we're going to make new data that preserves that variogram. So already this is a little bit different than the spin test because it's not preserving everything or just anything. It's very specifically preserving the variogram, or at any rate, that's what it's trying to preserve. And the way that you do that is you first shuffle your data randomly, and then you're going to compute the variogram of your shuffled data, and then iteratively tune or iteratively modify those data points. You're going to smooth this map such that the variogram looks as much like your original variogram as possible. And so, you iterate over k, and what is k? K is, there. K is the number of nearest neighbors that you're considering when you're trying to modify the value at your brain region. So, if K is really big, that means that the measurement at your region is going to depend on the measurements really far away, whereas if K is very, very small, you only care about the neighbors that are very close to you. And K is selected to minimize the error between your null variogram and your empirical variogram. And when you do this, you're going to end up with this null version of your brain map. Remember, you started off with the shuffled version, then you smoothed it in a way, iteratively, to make the variogram look as much like the empirical as possible. Now you have the surrogate brain map that preserves this autocorrelation structure. And what's really nice about this is that you can apply this to any sort of data. It doesn't have to just be the surface. This works in the subcortex. This works full brain. This works for voxel level data in theory, although it'll take a really long time because you're iterating. So this is already, you can kind of get an intuition that this is going to take more time than the spin test. Okay. Second version I'm going to tell you. So the previous one, that is the, here, let me go back. The previous version, this is Variogram-matching. You care about the variogram of your data. This came from Josh Spurt in 2020. And then we have this other version called Moran spectral randomization. So this actually came about the first time in the ecology literature in 2015. So this was not really hanging out in the brain imaging space until I think the first time it was used was 2020 in this paper, Vostevale. I don't really know how to pronounce his name there. And what's cool about this is, again, we're going to recap on Moran's eye. So if you remember, here we have Patrick Alfred Pierce Moran again. And this is the equation for calculating Moran's eye. And if you remember, one of the things I focused on was that distance matrix. That's really what makes spatial autocorrelation spatial, is that you take into account distance. And what Moran spectral randomization does is it takes advantage of that distance matrix, and it'll make new data based on that distance matrix. So what you do is you have your distance matrix. You're going to compute the eigenvectors of this distance matrix, correlate every eigenvector with your original brain map, and then, I'm just going to tell you a fact. You're going to have to take my word for it. This brain map X is equal to the average measurement plus the standard deviation times number of regions minus 1, and then times the eigenvectors of your weight matrix and the correlation of every eigenvector with your brain map. So let's just unpack this. Here's the spatial structure of your data, right? The eigenvectors of the weight matrix is telling you about space, and the correlation between your matrix and the eigenvectors of your weight matrix is telling you how much your brain map is spatially embedded in a way that matches this matrix. You see what I mean? So like maybe you look a lot like the first eigenvector and much less like the second eigenvector and a little bit like the third eigenvector, whatever. That's just going to tell you what your brain map's relationship with space is. So this is the fact. This is the brain measurement x is equal to this equation here, and we want a null version of x. So you can imagine we just need to like randomize one of these pieces and we'll be okay. And the piece we're going to randomize is actually this correlation between your brain map and the eigenvectors, and the way we're going to do that is like really silly actually, but this correlation, this is like, there's a bunch of correlations here, right? This is a vector, because for every eigenvector you're going to have a correlation with your brain map. The way that I'm going to randomize this is I'm just going to flip the sign randomly. So for every correlation we're going to randomly flip a sign. That's going to result in a wonky linear combination here, which is going to give me a null x star. And there you go, that's all it is. This method, by the way, is extremely fast, because the only thing you need to do is calculate the eigenvectors of W. You don't need to do any iteration, you don't need to do any tuning, you don't need to like find an optimal parameter k that matches blah, 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 blah. And also it's going to give you identical Moran's I, and it's Moran's spectral randomization as your original brain map. Again, code, here's how you do it. Okay, final version I'm going to tell you, I'm like at the very end of my talk now. This is very, very new. This came out this year. It's a preprint, 2024. If you remember last year, there was that Nature paper that came out about the eigenmodes of a surface. And the idea here, just as a refresher, is that you have some surface mesh. This is not data, right? There's no empirical data here. It's not a brain map. It's a surface. You can calculate the modes of this structural object. And so that's what these things are. And then so if you do have data, let's say you have some brain map Y, you can recompute, or you can express your brain map here as a linear combination of these modes. So you're going to find some optimal betas for each of these modes to recreate your brain map. And these modes represent the space, literally the structure of your brain. And so you can imagine if you want to randomize this, what if we just slightly randomize these modes in a way that is hopefully clever, and then recreate a null version of this brain map. And so the way that this is done, introduced by Nikitas Kousas here in this preprint, very recent. And how you do this is that if you remember your spherical harmonics, which I don't because I never learned about spherical harmonics, but what you do is you have the modes of your surface are going to come in groups. And these groups you can treat like you have three, like a mode is a vector, right? So if you have three vectors, you can imagine that's a sphere. You can just rotate that. We've already been over the whole rotation thing. I don't need to go over that again. And so because you have these groups of modes, you can rotate them in a way that gives you these slightly modified looking modes. Not too different, but they're just a little bit different that you've created from this rotation process, but you're going to keep the betas the same. So instead of your original brain map Y, you're going to have a brain map Y star that preserves a lot of the spatial structure of this brain map. It preserves the betas, right? So depending on your brain map, how much is the first mode contributing versus the second mode versus the third mode, etc. That's going to be preserved, but you're just like modifying, you're rotating these modes in groups. That's what's happening here. And just to like really quickly wrap up, now I've told you a bunch of different ways. Like this is a previous slide about, why do I want to even bother with all of this? Well, the point was you want to be able to create these null brain maps over here. And now we have many different ways of creating those null brain maps. So depending on your situation, you're going to use a different one. So yeah, we now know a way of making Y null. And just to show you what this looks like in the wild, here I have a bunch of correlations for a bunch of different brain maps. Then I make a bunch of different nulls for all of these correlations. So for every correlation between two brain maps, I'm going to get 1,000 nulls, or 10,000 or however many. And then I'm just looking at, for each correlation, how many are statistically significant, or sorry, across all correlations, how many are statistically significant. And what you see here is if you take the spatially naive models, the ones that do not account for spatial autocorrelation, a lot are statistically significant. And if you use spatial null models, way fewer statistically significant. Here it's a comparison across the different null models. This is just 2 different atlases, so you can see it's quite reproducible. And if you zoom in on this, you can see that some of the most conservative, that version I showed you at the end, Moran spectral randomization. Eigenstrapping is not here because it's very new, so it just hasn't been incorporated yet, but Moran spectral randomization, that is very conservative. The variogram-matching is less conservative, and then the spins are usually somewhere in between. The point of this is just that spatial nulls work. So they do account for the spatial properties of your brain map. Now, I also just wanted to take a second to let you know that this is not just for correlation. So a lot of my examples are always correlations, because correlations are really easy to talk about. Everybody knows what they are. You kind of get the intuition. But you do not need to use spatial nulls only for correlations. They work for all sorts of things. So, for example, you could do it for a network enrichment where, let's say you have some brain map, some phenotype, let's say metabolism of something. And you want to know, do I have more metabolism in a specific network? So like the visual network or the somatomotor network or whatnot. So what you can do in these cases is that you calculate what is my metabolism in the somatomotor network, and then you generate all these nulls and ask, and then you can make a null distribution of the metabolism in all these null networks based on the spatial null models. And then doing that, you get a sense of which networks do you have enrichment. Statistically, like enrichment that is statistically significant against the spatial autocorrelation-preserving null model. So this is just an example. Spatial nulls can also be applied to network enrichment analyses. And then just final example that spatial nulls can also be applied to correlating matrices. And so here's a matrix. I'm going to correlate the upper triangle of this matrix with the upper triangle of a bunch of other matrices. That's what these correlations are. This is work that I did a couple years ago. And the way that I got this piece spin here is that I rotated the rows and columns. Now, you should be saying to yourself, this is a bit weird. How do you do this? Because now you have a matrix, not a brain map. So how are you supposed to spin the brain map that's not a brain map, right? But I did a really sneaky trick here, which is that when I wanted to create when I do my spins, instead of spinning the data itself, I just asked it to spin the indices of my brain map. So you can get a sense of how to shuffle your rows and columns in a way that preserves spatial autocorrelation. That'll give you that p spin right there. So the point here being that spatial nulls, or the spin test specifically, can be used to apply rotations to region-by-region brain networks. So the generative methods aren't going to work here because the generative requires that you start with actual empirical data, but the spin test works because you can just rotate the indices. This is actually really, really convenient if you're actually using this in your work. Spinning the indices is way faster than spinning the data by itself, because you can imagine if you spin the data, you have to do that for every single time you have a new brain map, whereas if you spin the indices, you can do it once and then apply that for anything. Just sort of life hack there. Okay, that's all. Resources, so this is where a bunch of the code is. I was showing a lot of code from Neuromaps, which is a toolbox, and I was showing a lot of figures from this paper here, 2021, comparing spatial null models for brain maps. I strongly recommend, if you want to know when to use which, hopefully you kind of got that sense from this talk, but when to use which null model and what's important for your data and whatnot. Good, thank you. Questions? Yeah? One or two? Yeah?
You showed from the Markello 2021, it shows that all these methods have inflatable forces. And if you look at the Goose paper, they also show that the Moran method has those.
Mm-hmm.
So I feel like, do you mean that you shouldn't use it?
Sorry. Yeah. So the thing to remember here is that we're preserving spatial autocorrelation. That's one thing we're preserving, one relationship. But brain regions have all sorts of relationships. And so if you don't preserve for anything, those inflated false positives are like insanely high. Like, you know what I mean? Like the number of inflated false positives are super high. Spatial autocorrelation preserving null models brings down how many false positives we have by a lot, but it certainly is not perfect, because you can imagine just by preserving one thing doesn't mean you're preserving everything, right? There's probably all sorts of other things that are going to impact. And so yeah, I totally agree. It's not perfect, and you're going to need more things, but I definitely think it's better than using nothing, because it's way less than using nothing.
Well, it's better than doing something terrible,
Yeah.
But it's still pretty bad, right? I mean, people use it, they'll get results.
Sure, yeah, for sure. And that's where things like multiple comparisons comes in, or having to use a null model depending, for example, if it's time series data, maybe you need to use a time series null instead of using a spatial autocorrelation-preserving null. I mean, I think the spatial null models preserve spatial autocorrelation very well, especially the Moran spectral randomization. And that's what they're designed to do, but trying to beat, I mean, at some point you're going to need what postdoc in the lab, Andrea, says, the everything preserving null, right? Like that's sort of the gold standard. One more question? Yeah? We're good?